Hey everyone, thanks for coming to today's video lecture. We're going to be getting into Unit 4, Chapter 15, Adaptive Immunity, and how the adaptive immune system creates immunity to specific pathogens and a memory to protect from future pathogens, and how we can take advantage of this with immunization to make sure that when we do get infections that they are not as severe as they were previously. And with that, let's get started. And so the adaptive immune system is that third line of defense. First, we have those first line. So first we have those first line of defense, which is those physical and chemical barriers that are non-specific and ever present. We have the second line of defense, which is the cellular response that is non-specific to pathogens within the body. And then we have the adaptive response. And the adaptive response is an acquired response that is shaped by current and past infections to fight infections. And so it uses this by detecting and utilizing antigens, which are detected by these antigen receptor proteins, which we'll talk about are T cell receptors and B cell receptors or antibodies. Excuse me. And antigens are kind of pretty much anything that these antigen receptors are able to recognize. These receptors are capable of recognizing countless antigens, and so it's it's a fairly broad category of thing to consider an antigen, but not everything is the same level of antigen. So there's antigenicity. So things can be more antigenistic than other things. For instance, there are size requirements. And so at lower than 10,000 molecular weights, you kind of don't have that antigenicity because the antibody or the T cell receptor aren't able to like recognize the molecule. And so when you think about a whole protein, you have this whole protein and you have this 3D surface, you have that 3D shape, and that 3D shape has a 3D surface. And that 3D surface can be recognized by these antibodies anywhere on its surface. And each of those locations that can be recognized are what's called an epitope. And so you can have one protein and it can have many different epitopes on it, but a particular receptor is only going to recognize one epitope. So then we have, like I said, anything can really be an antigen where you have all of these different kind of biological molecules. You can have proteins and you can have lipids, and you can have like some complex molecules. Now, one thing that is important is that one thing that's big with the immune system is differentiate. Excuse me. Is this one thing that's important with the immune system is differentiating the self from non-self. And so things like pure DNA aren't very antigenistic because they're found over large places, like large slots of life. Those things that would also be true for like receptors that are seen across many different types of not only like animals, but like bacteria as well. So you can have antigens that are on the self that are specific to specific genetic backgrounds. So specific portions of populations may have specific antigens or specific receptors or proteins that are just slightly different than the rest of the population, and so that epitope can maybe have different antigenicities and different antigenicity profiles. And so these things are something that's very common. Is like blood grouping is one that's very common. So then you have kind of like these super antigens, which are these molecules that are able to stimulate T cells in a really robust way, and they're able to release a ton of like cytokines, and that recruits more cells in a larger immune response, and it's kind of like a snowball effect, and it causes damage more than just like a normal infection would, because you're actually exacerbating that damage with this response because it's kind of overreacting. You then have allergen, where allergens are these molecules that aren't yourself. So they're non-self, but they're not pathogenic. But because they're non-self, you can recognize them as non-self, and you're saying, hey, this is foreign, where it's problematic. And so you have like antibodies, IgE, that can recognize those and create this allergic response to these foreign molecules. And then you have autoantigen, which is going to be something that is the self. Essentially, it's just like anything that you would find on yourself, in your, like, not on yourself, but like in yourself. It like it is you. Autoantigen, it would be essentially any of the proteins that are supposed to be on the surface of your cells or circulating within your body. Those are autoantigens. And then you would have like an autoantibody that recognizes that and cause problematic behaviors, because then you're trying to attack yourself. But typically the immune system has selective mechanisms that eliminate those receptors that can recognize these autoantigens. And so in the development of B cells and T cells, there's these positive and negative selections that kind of make sure that we don't have receptors that can recognize these autoantigens. For instance, this blood grouping would also be considered autoantigens. There's kind of some overlap with these terms. So something that's really important with this immune system is being able to recognize pathogens that we have encountered in the past, and being able to adapt your response to a particular infection that you have currently. And so the things that differentiate this adaptive immune system is the specificity of being able to identify and drill down and like respond to this one particular pathogen that's a problem currently right now. And then we can also remember that pathogen for later, and that's called memory. And we do this by kind of stowing away cells that kind of would recognize that pathogen, so that in the case that we see that pathogen again, we can kind of unpack that cell, like very figuratively unpack that cell. It's like in the body somewhere, and it will be activated. And then that can respond to the pathogen when we interact with it again. And so these immunities can come in B cells and in T cells. And T cells facilitate this like cell-mediated immunity, while B cells facilitate this humoral immunity, which is more of like your blood. And so your T cell-mediated immunity is your T cells are kind of monitoring the cells for normality, and making sure that your T cells are, your cells are expressing only self. And if your cells are starting to express things that aren't self, that's kind of a red flag that there's something problematic, and these T cells can go into action and likely kill or tell the cell to kill itself. While humoral immunity is going to be like just like in the blood and in the interstitial fluids, in the lymph, and in the mucosa. And these are the things that are going to be like antibodies that are just being secreted out and identifying pathogens that are just like hanging out in the tissue, hanging out on like linings of things, hanging out somewhere like that, not inside the cell, like intracellular parasites, intracellular microbes. Those things will be a T cell response, while these like intra, which would be like in between the cells, pathogens would be like more of a B cell response. So you have the immune system, and this immune system has kind of originated from this common lymphoid progenitor, which in the last video we talked about how it develops from a hematopoietic stem cell. The hematopoietic stem cell is in the bone marrow, develops into all kinds of innate immune system cells, but also it develops into this common lymphoid progenitor. And this common lymphoid progenitor can migrate into the thymus, or it can stay in the bone marrow. If it stays in the bone marrow, it gets committed to the B cell lineage. If it gets into the thymus, it gets committed to the T cell lineage. And then you have early developing T cells and early developing B cells. And those cells develop into innate B cells, and the T cells develop into, I don't remember the name for them right now, but I will get to it later. So, but either way, after development, and these, they go into the lymph node, and they circulate through the circulatory system in the lymph system into these lymph nodes, and they kind of hang out in these lymph nodes until they are activated. And so kind of what happens within the tissues is you get this pathogen that comes in, and your cells are going to recognize that there's this pathogen. And there are these systems within your cells. All cells have it. They'll just, sorry. You have these dendritic cells and these macrophages, which are these professional antigen presenting cells. You have like antigen presenting cells that are like in all parts of your body, but we'll talk about like the professional antigen presenting cells currently. So you have these dendritic cells and these macrophages. And they will recognize that there's a pathogen and uptake that pathogen and then digest it into tiny little pieces. And then once it's in tiny little pieces, it'll put them out onto its cell surface and say, hey, look at all the things that I found. And a T helper cell, which is a lineage of it is a developed T cell, and it will go into and it will recognize these MHC complexes and be like, oh, that is problematic. And it will start going through this like cellular pathway of like responding to this antigen, because it's now saying, hey, something's going on. There's now a foreign material. So essentially like this dendritic cell is kind of like this like center, like a centurion? No. Those guards that like are stationed. Just it's a guard. And so it's just surveilling the area, and it is saying, oh, something suspicious is happening. And now you have its manager essentially comes by and says, oh, you're right. I'm going to sound the alarm. And then it goes and starts that process essentially. And so that T cell helper cell is activated, and it can be differentiated into cytotoxic T cells or memory T cells or different kinds of T cell helper cells. You also have T helper cells that can activate B cells, and those B cells can then have that humoral response with those antibodies. And those antibodies, that B cell can be activated, and it can differentiate into plasma cells or memory B cells. That memory B cell is where we're going to get that memory and long-term storage, where plasma cells are a secretory cell where it takes antibodies and it secretes them out into the environment, and it does so in a really high-paced environment, really high-paced kind of like rage out kind of lifestyle, and so it's really short-lived. And so plasma cells are kind of like, we really need these antibodies right now, right now, right now. And then once we don't need those plasma cells can go on and apoptose, go into cell death. And then you could have then you can have memory B cells, and those memory B cells are going to be like those more long-lived. Those are going to circulate through the circulatory system and the lymph system, hanging out in the lymph node primarily, waiting to be activated again when that antigen is encountered for a second time. So we have the T cells that are recognizing these different cells. You have the like professional antigen presenting cells, and then you have like non-antigen, sorry, like non-professional. That's not the term, but like you then have just like regular antigen presenting cells. And so, sorry, let me get back to it. And so those professional ones are using MHC2, while the non-professionals are using MHC1. And so MHC1 is found in all of your cells, and so your T cells are able to like survey all of your cells to make sure there's nothing just like awry with them, and it uses MHC1. MHC2 is used by dendrocytes and macrophages, and those are recognized as like, these are currently problematic like pathogens. And so it's kind of like two separate signaling pathways. But they both can activate T cells, but they do so in different ways. But anyway, so then you have different kinds of T cells that kind of help with this response. You have T helper cells, which are this primary response to professional antigen presenting cells. And they help kind of recruit this immune response and initiate this immune response. You have the cytotoxic T cells, which are the ones that kind of survey your tissues. They're the ones that kind of look at all of your cells with the MHC1 complexes and things and see, is this cell completely the self? And if there is one that is not fully the self, it is killed. So then we have natural killer cells, which go through and do similar things as the cytotoxic T cells. And so we have those, the CD4 T helper cells, and those that's stimulated by the MHC2 complexes that are by these dendritic professional antigen presenting cells. And that signals this differentiation into different kinds of T helper cells, and those differentiations occur based off of the different cytokines and signaling molecules and complement signals that are being, not complement signals, coupled signals. Give me a sec. Co-stimulation. So these cytokines and things, there's depending on the co-stimulation of this T helper cell by different signaling molecules, it will differentiate into different T helper cells and have different responses. You then likewise have the cytotoxic T cell, which can survey the TCA cytotoxic T cell, which has the MHC1 complex. It recognizes the MHC1 complex, which are found in all of your cells in your body, and those can be influenced and recruited by T helper cells, and they can recruit memory B cells, and they can eliminate the cell that is affected. I'm sorry, they can differentiate into memory T cells, and then they can also be toxic to the cell that stimulated them. And so it causes them to go into a self-destruct cell process. So when superantigens are, we talked about how superantigens are like when there's overstimulation of this process. And so it can cause a like vast overstimulation of these cytotoxic cytokines, and it can cause damage to the tissues, and it can cause toxic shock syndrome. So then we have the humoral immunity, which is like antibodies and the B cells and immunoglobulins. Sorry, immunoglobulins. Um, and so we have our naive B cell that is inactive, and it is before it's been uh, before it's been exposed to a microbe, and it's just been exposed to a microbe, and so now this is a non-naive B cell, and so essentially it has all of these antibodies all over its surfaces, and this is most likely IgM. IgM is the first isotype of antibodies that are produced, and it can recognize some kind of epitope or antigen on that microbe or pathogen surface. And that can cause this B cell to need to be activated by a helper T cell. And this goes into and this causes it to be an activated B cell. And this causes it to start to differentiate into a memory B cell lineage, which is that long-term storage. Or we have that plasma cell, which is that kind of like a factory for antibodies. And so, and then it produces those antibodies out into like the blood to fight off those microbes that we originally detected. So it uses antibodies, which are shaped like this, and they're shaped like this, where you have this is a peptide, which is one protein. You have this here, which is a second protein, and then you have that mirrored up again. So you have 4 peptide chains, but 2 unique sequences, and those proteins are conjugated together by disulfide bonds through their histines. Nope, sorry, they're cysteines. There's, sorry, this SCYSINE, cysteine. Lord. There's cysteine residue, which has a sulfur ion. Those are able to have a covalent bond together and create a disulfide bridge. And that's able to like essentially lock these peptides together. These peptides are also being held together by protein-protein interactions like van der Waals forces, hydrogen bonds, and things like that. So then you have 2 different regions of the antibody. Well, okay, you can kind of think about it in a few different ways, but you can think about it right now in 2 different regions where you have the Fab and then you have the FC, where the Fab is this fractionable or fragment antigen binding or antigen binding fragment. And that's because your antigen binds up here. You then have the one that really makes more sense, which is your, well, both of them make sense, but this one is your crystallizable fragment. And crystallizable is a bit strange, but it is because of X-ray crystallography, which is a technique where you're able to take a protein. Well, you can take any compound, but in this case, we take this protein and you kind of you make a crystal of it where you have essentially made a really high concentration of the protein, and you've concentrated it down to such a degree that it crystallizes, and it crystallizes in like a very ordered fashion, hopefully. And what early researchers found a couple of decades ago is that this bottom region here is very easy to crystallize. And when you have that crystal, you can then shoot X-rays at it. And depending on how the scattering of those X-rays, so you shoot an X-ray at this crystal and it will scatter depending on the crystal itself and the internal like lattice of the crystal. And all that to say, the shape of the protein makes the crystal do different things, And the X-ray, when it hits it, will scatter differently based on that protein's shape. And so you can then say, oh, well, this is this diffraction pattern that was found. And you can back calculate it using really complicated math and to say, this is the 3D structure of this protein. And this is how we as scientists know the shape of so many of our proteins is through X-ray crystallography. And currently we now have cryo-EM, which is a different type of method of finding out structures, but early on, X-ray crystallography was the best way, and that's how we solved actually the structure of DNA. Anyway, so this bottom region, very easy to crystallize, and so it's called the crystallized one fragment. Then we have this antigen binding fragment, which did not crystallize very easy, and that's partially because it's just a little bit more loose with its structure. And then this part up here is variable, depend on every cell is variable, and so that makes it a little bit more difficult to get something that's constant. So anyway, this antigen binding site is different for every single B cell. And though the shape of that binding site is what determines what antigen it is able to bind to and what pathogen essentially it's able to recognize and neutralize. And so these antibodies can be secreted out and then recognize and do a couple of different things. You can recognize pathogens, and it can be used to stimulate what's known as the complement system, which is this like really large complex of proteins and that like come together when this pathogen is present with this antibody and a few different other considerations that I'm not really aware of. And it punches a hole through the plasma membrane of the cell. And so that punches a hole through it, and it causes the cell contents to rush out and the environmental liquids to rush in, and it causes the cell to essentially die. You then have optimization, opsonization, where you've essentially coated the bacteria to make it more favorable for macrophages to phagocytose it, whether that be it's making it easier to phagocytose, whether that like because it can now recognize what it needs to swallow, right? So the macrophage is recognizing the fractionable, the FC regions, and it's able to better engulf those microbes. You then have neutralization, which is kind of the same thing, where it's able to cover the surface of a of a pathogen in such a way that it isn't able to do what it needs to do. So for instance, if a virus needs to make contact with your cell to get into it to make a problem, if you have an antibody that blocks that virus from recognizing that in some way, I don't have three hands, but like this, you have your cell receptor and you have your pathogen that can recognize and it can go into your cell. If you have an antibody that can recognize that, it won't be able to get in or recognize it, right? And so it causes it to neutralize. A very specific kind of this neutralization is like antitoxins, where your antibodies is able to bind to these toxic proteins that can do harm to your tissues, but it can neutralize them where they're not able to do the function that they would otherwise do to cause harm. So we talked about that FC region and how that FC region can be like bound by the macrophage to help with like phagocytosis. And then we talked about the complement system. And the complement system, if you I would highly encourage you to look up an animation of the complement system because it is highly complex, and it's essentially all of these molecules, all of these proteins coming together in a very like systematic way. And it's also a signaling pathway so that like one protein is activated and it causes another protein to be activated and it causes another protein to be activated. And those activations causes like are sometimes catalytic, where proteins are like broke apart and where one's now active now that it's broken apart. And sometimes it's like a shape change, all kinds of things. But overall, when this pathogen is around and an antibody is present and the conditions are right for the classical complement pathway, there's a couple of different complement pathways. There's the classical, there's the lectin, and then there's the alternative pathway. But anyway, these many different proteins all come together and they form this pore in the membrane of the pathogen. And this pore is allowing liquids to come in and out of the cell freely, which is problematic in keeping up the homeostasis of the cell. Because you can't keep up chemical gradients when you have these kinds of holes. And so it just inevitably leads to the death of the pathogen. We talked about how immunoglobulins have, I mentioned just a little bit that IgM is the first kind of antibody that is produced by innate naive B cells. But we have multiple different kinds of classes of antibodies, and that fractionable region I keep saying fractionable region, that crystallizable fragment is kind of what makes up this constant region or the isotype region. And so you have this region here, this little leg. And you have different versions. You have IgM, IgA, IgG, IgD, IgE, and each of them kind of have different functions and different roles, where IgM is the first ones that are produced, and then eventually through different responses, IgG and the other ones are produced, where IgG is monomeric, IgA is dimeric, IgM is pentameric, IgD and IgE are also monomeric. IgM is that first one, and it's what is going to be used by your naive B cells, and then eventually they will differentiate and they will class switch into IgG after stimulation of an antigen. And then also IgE is important with allergens, and so they are important in your allergy response and also your response to pathogens. You have IgA, which is a dimer, and this is a antibody that is specifically secreted out in your mucous membranes. And so you can think about IgAs in your nose and your mucus there, and in your gut, for instance, would be another place that you would find IgAs to help with that line of defense. So the thing that makes your adaptive immune system different is that it can remember past infections for next time. And so like a key mark of this would be that the first infection, the response to the first infection is different than the response to the second infection. And so you can think of when you have that first infection, you have this latency period where the you can think really zoom in. Your pathogen is attaching, it's starting to proliferate, it's going through its like the like stages of infection. Then your innate immune system is going to start realizing that this infection is happening and starting to respond to this. And so your innate immune system is a lot faster because it's non-specific just to like any old pathogen. And then you have the adaptive immune system, which is a more specialized response to that pathogen. And it takes time for that specialized response to actually happen. And what we see is that if you looked at the blood titers, is that IgM is the first response, and it is the majority response for that as that primary response. And then you have eventually class switching that has occurred and more specificity that has been gained as these B cells have differentiated, sorry, not differentiated, but they've developed into IgG isotype B cells. And so then that IgG response is that major responding antibody type. And then eventually your infection is going to be satiated and it's going to be resolved. So then some of these B cells are going to be shifted. Some of them are going to be plasma cells, and so they're just going to die off. And then some of them are going to hopefully be memory cells. And if there were memory B cells, they will be in your circulation until next time. And that second response is that next time. And so you have that infection, that pathogen infection, and then your innate immune system occurs still. And then a couple of days later, a lot faster than the first time, you're able to kind of recruit or like bring back from those memory B cells your response, and you're able to respond much faster than you were the first time. And so you're able to do this where you have IgG as one of the more majority of this response rather than the IgM. You have the IgM, which is that still the same kind of first response, but the IgG is those like memory B cells. It's really coming from those memory B cells. So something to think about is that when you do immunization, you're kind of skipping this first response and going straight towards the second response. You're kind of you're priming the adaptive immune system to say, hey, remember that this is a pathogen for next time, without actually having to go through the actual sickness of it. And so we can think about our immune system in a couple of different ways. Like we can classify our immunity in a few different ways, where you can have active and passive immunity, where you have active immunity is a long-lasting effect. That is something that's like the cells produce antibodies, and those antibodies stay in the person. Then you have passive immunity, which are just like not long-lasting, where the antibodies are kind of introduced and then they go away just by natural like degradation. And then you can have immunity that is naturally occurring, or you can have immunity that has been artificially produced. And so you can kind of think about this in a few different ways, where you have like active natural immunity, where naturally occurring active immunity. So this would be just like regular, what we've pretty much been talking about, that a pathogen has stimulated the immune system and you create a natural response to that and then a memory response to that. Just kind of live in your life and it happens. You have passive immunity, which just, again, just naturally happens, but instead of having the cells to produce the antibodies yourself, in this case, how would you be delivered just antibodies by themselves? Something like this would be through the placenta or through breast milk, where they're delivered and you're able to kind of leverage them for a time, but eventually they'll disappear. And so they're not permanent, where these active immunity that you've got is more permanent. You then have artificial, where you could have artificial active, where we've stimulated these cells and stimulated this response, like with a vaccine, or you can have passive artificial immunity, which is something kind of like blood transfusion or like antibody treatments. Let's see, yeah, antibody treatments. And so here you can kind of see a breakdown a little bit with nice photos. And so now we can talk about how vaccines kind of leverage this immune system and this immune response to specifically kind of stow away responses for in case we encounter pathogens in the future. And so, yeah, so like the definition, deliberately exposing a person to a pathogen so that they can be like affected, they can be like protected from that pathogen in the future. And so there's a few different like things to think about when it comes to vaccines is that they need to not be very toxic and they need to not have very many side effects and they need to be able to protect against pathogens that we would regularly encounter that would cause us infections. We need to recognize that that these vaccines should these vaccines should should activate both B cells and T cells because both of them are needed for that adaptive immune response, and that we want the response to be long lasting. We want them to go into like a memory, so that we can use them for later on. Ideally, we wouldn't want very many boosters. We would want it to be such a good memory that you wouldn't need to remind the immune system essentially that it exists, and you'd want to be able to produce it cheaply. And vaccines can be made in a few different ways. You can kind of think about how we have this immune system that that is stimulated by pathogens. And for us to say, hey, there's this pathogen, please remember it for the future. How could we do that? And it could be just giving it the pathogen itself and says, hey, remember this. And that would just be like getting someone sick intentionally. And there's ethical concerns with that. But it would, in fact, cause the immune system to be stimulated. It would cause an immune response, and it would cause there to be an immune memory to that particular pathogen. But it's dangerous. And so what they've done is they've found that we can mutate, or there are variants of these viruses that are not very deadly, or they're not very infectious. And so we can kind of use them to take place of the like really deadly one, right? So they look the same, but they're just not as deadly, right? And so this is kind of the whole idea of having a killed cell or an inactivated cell, inactivated pathogen. And so you could have um, like a bacteria that you've like killed all of like killed it, but all of the like the cellular components, all of that are still there. And so all of those surfaces, all of those epitopes, all of those antigens still exist. And so you might be able to use just dead corpses of bacteria or pieces of viruses to stimulate that immune system, right? Or you could have the attenuated viruses that are like not as deadly or not as pathogenic to the cell. You can then also just like give the molecules themselves, just like break off a piece and say like, look, here's a little tiny piece of the pathogen, please make an antibody towards that and then remember it. And then we can talk about how genetic engineering has kind of like made it where we can package this up into a much like more stable and a much more like cost effective and a lot more cool system. So we talked about how you have those just having a killed pathogen and injecting just the killed pathogen. So something like the cholera or the polio vaccine or something like this. We talked about the attenuated vaccines. These things are like mumps, the measles, mumps, and rubella vaccines. You can have pieces and components of the bacteria, like specific pieces of a toxin, for instance, or very specific antigenistic molecules that have been like studied and found to be really important for immune responses, and they can then bottle that up and then inject that, and then that will be like, oh, we can recognize that that is that little piece of that pathogen later on, right? And so now there's a little bit better like methods of like there's newer methods of introducing these like antigenistic proteins into the cell. And we can do this with like recombinant DNA or mRNA. Sorry, those aren't the same thing. Those are recombinant DNA or RNA. Both are different approaches that can be used where you can inject some kind of plasmid that has some gene, and that gene might have essentially the antigen that your immune system is going to need. You can deliver that into a cell, and that cell is going to then make the antigen for you. So then you don't have to make the antigen, and you don't have to inject it into the person. You just have to make this DNA, and that can do the work for you. And because DNA is fairly unstable, once it's in the body, it's going to be degraded fairly quickly, and you're not going to have it like lingering around. Now, RNA is even more unstable, and so it's going to be degraded much faster. But it also doesn't have to go through so many machineries. So something about DNA is you have to get it into like the nucleus, because you have to go through that central dogma of DNA to RNA to protein. And so for DNA to go from DNA to RNA, it has to go into the nucleus and be transcribed by tag polymerase, by polymerase, not tag polymerase, by polymerase, and then by DNA polymerase. And then mRNA, on the other hand, doesn't have to be delivered into the nucleus. And so it's just like a little bit easier. It's a little bit fancier because it just doesn't have to do that. So because it skips that DNA step, it goes straight from that RNA, it can go RNA protein. And so you can package up this mRNA to be delivered into cells. And this package is essentially protecting it so that just it doesn't get degraded, because there's going to be enzymes within the interstitial fluid and extracellularly that can degrade DNA. It can degrade mRNA as well. And so packaging it up in these little lipid nanoparticles can help you protect that cargo. Additionally, you can add things like receptors and specific compounds on top of or inside these like packages or these vesicles. And it can make it where you specifically target specific cell types. And so you can like target, oh, I want to deliver into this very specific cell type. You could do that theoretically. But usually you're just, right now, we're just going into the cell, putting in your mRNA, it's going to the ribosome, it's getting transcribed into proteins. Those proteins are just little pieces of antigens that are important for your immune response, and then we can use those to elicit an immune response and kind of bypass that first response and kind of go straight to the second if we ever do actually get infected with the pathogen. So we kind of talked about how we kind of bypass that DNA to RNA by going RNA to protein, which is that central dogma of biology or molecular biology. And then another thing you want to think about with vaccines is kind of why they work and why they're important. And so we can kind of think about having an infected person around, and if everyone around them is protected, then you don't have this risk of someone becoming infected, right? And so for instance, let's say these dull dots are unvaccinated people, and these purple dots are vaccinated individuals. And we're going to make a rule that only can you be infected if you touch an infected individual. And so, like, oh, someone got infected. Oh, someone got infected. I'm going to count that's really far, but oops, someone got infected. Oh, someone got infected. Oh, you could say someone's vaccine broke and it wasn't strong enough for some reason, or maybe they like actually inhaled so much of the pathogen they just like fucking drank it, you know? And so they actually overcame the immunity that they had and they got sick. But what's interesting is the person that isn't vaccinated, but they're surrounded by people that's completely vaccinated, they're protected and they're not going to be infected. And that's what the idea of herd immunity is that because there is a majority of people that are vaccinated and have this immunity for a specific pathogen, we can protect those who might have some kind of predisposition or some kind of problem that might make it where it's fairly difficult for them to become vaccinated, whether that be that they are autoimmune, and so vaccines can cause some more um, like something that immune issue. So vaccines are foreign materials entering into the body. You're entering foreign materials into the body. What is a non-specific response to foreign materials is the second line of defense, which is that innate immunity. And that innate immunity, a part of that is inflammation. Um, and inflammation is a big part of autoimmune disorders like rheumatoid arthritis and things like that, and joint inflammations and things like that. And so kind of thinking about um, how having a foreign material in your body can create that innate immune response, and that innate immune response might exacerbate someone's like medical conditions, and it might disparage them from getting a vaccine where they can't get it. And so having a lot of people that are vaccinated is really helpful and beneficial and protective for them because they are at a disadvantage otherwise. And so it gives them that kind of just like the right thing to do of just like giving them the protection that they can't afford themselves. You know what I mean? So anyway, so then we have all of these vaccines that are required by Oklahoma. So these are all of the vaccines that are required before entering into childcare. Hepatitis B, that is a sexually transmitted disease, but it also is passed between mother and fetus, and so in mother and child during like childbirth and things. And so vaccination and early vaccination for this is really important so that that virus doesn't take hold. And hepatitis is I believe it's a retrovirus, and so it integrates into the genome of your cells, and so it's a permanent kind of thing. Is once you have it, you can't really get rid of it. And so for someone who's an adult, when you get it through sexual intercourse, that's preventable because you don't have sexual intercourse, or you have protection. For a fetus, when they're given birth, when they have been given birth, they don't have any protections against that. And so they can be potentially exposed if the mother is positive and is shedding viruses at the time. And so having a immediate vaccination for that is very important. You also have Tdap, which is diphtheria, tetanus, and pertussis. And this one's really cool because it has multiple different it protects for multiple different pathogens within the same vaccine. So it's kind of like a 3-for-1, And so one thing that I've heard before is kind of like, why don't they just make there to be less vaccines? And one is probably vaccines are just really hard to make. And so making a vaccine that's mixed of multiple different things is even harder to make. But they do that. They definitely do that. Measles, mumps, and rubella, the MMR vaccine, for instance, is another one that does that. And so here's another set of vaccines that are important once they go into grade school. So I talked about that already. And then with that, that concludes our chapter. So I hope that was really informative. I hope you enjoyed it. I'll see you next time. Bye-bye. Oh, hello.